Hi, this is Miss Litton, and this is the APBio Unit 5 review. Say hi.
Hi.
We really have reviewed everything, so it's going to be more like, do you have some questions and things that you want to go over? I will tell you a little bit about the test so you're prepared for the test. So for the essay portion, I asked each class 3 questions. Okay? You can be sure that one of those 3 questions is going to be a highly suggested reading and thinking, right? And so what you need to do is you need to open up your book, right? And I've already essay tested you over a large portion of that, right? All those chapters. So your highly suggested reading and thinking is just got to come from the remaining, 3 chapters? So that's going to be it for sure. It's not like, I wonder if she'll ask us. Okay, I'm going to ask you 1 or 2 highly suggested reading and thinking questions. So be prepared for that. Then you just need to look through and think, what are the big ticket items that she hasn't asked us? We already went over the essay review, everything, so I would look over that because that would be a way to prepare for that essay. Don't get so where you memorize every little thing, you don't have a big idea of what you're really talking about. Like when I asked you, how does this increase fitness? How does this help with that process? Look at your big ticket items and go through them. What we spent most of our time on in the class, that's probably what I'm going to ask you about in the essay, besides the essay prep that I already did. Okay? The multiple choice, because you're going to have more time, because your essay question's so short, I ask more in the multiple choice only because that's a benefit to you the more questions I ask you. So normally, how many questions do I ask you?
25.
I ask you 25 questions, 2 points a piece, and that's what? 50, right? So I ask more. I can't remember, 44, 46, 48, somewhere in there. Okay? I asked you more questions, and then I think I have to multiply it when I did the math, like by 1.7 or something like that, in order for it to be still 50 points. But now, every question isn't worth 2. It's less than 2, and that gives you, if you miss one, it's not as penalizing. Does that make sense? Okay? And then you just have to, you're going to need to approach, you've got a lot of chapters, right? 8 chapters. And last time I reviewed with you, we talked about ways to break that down. And so if there are things that you want to talk about, if I say now. Sorry, I heard that song this morning. Let me get out my calendar real quick so I can look at it. If you look at the first thing that you could have multiple choice, and again, they could be in any order, so I don't want you to think that this is the first question I'm going to ask you. But if we look at our calendar, which I seem to be unable to do, I think 18 is your first chapter, right?
Yeah.
Okay, so if you look at chapter 18, it's the origin and history of life. Now, besides the essay questions, which you need to prep for, when you think about the origin and history of life, things that come to mind when I think of that chapter is I think about early Earth, right? And I'm not going to redo everything. I'm just kind of hitting a few big ticket items here in chapter 18. I would know about early Earth. Right? What were the conditions of Earth? Are they different than the conditions now? What were the steps that life had to go through? What were the different things that you had to do? Things like you needed to make organic molecules, right? You needed to make those bricks. You needed to make polymers, right? You needed to make a protocell. And then you needed to make a cell. And in what way, what steps did they, all good? Good, okay. In what way, what steps had to evolve to get them there? When you first saw it, did you know who that was? Yeah, old VP. So when you had a first cell, do you think your first cell was a prokaryote or a eukaryote?
Prokaryote.
Probably a prokaryote, right? Because eukaryote's more complex. You can think that through. You think your first cell was an autotroph or a heterotroph? Because it was easier to acquire nutrients. Then, was the first autotroph doing cyclic or non-cyclic photophosphorylation?
Cyclic.
Probably cyclic, no oxygen available, right? Then, once oxygen is in the air, right, then what kind of respiration can you do? Not anaerobic, but aerobic, right? So just make it make sense to you as you're processing that, right? So we talked about the formation of the early Earth. We talked about eventually eukaryotes. Am I hitting all the main things in that one? I think that's it, right?
Yeah.
Okay, what's after chapter 18?
19.
Chapter 19. All right, yeah, how about that? 19 follows 18. So on that one, you were looking at taxonomy. You were looking at, hey, here's all of life. Maybe this time, know the organization, how it goes. Domain, kingdom, phylum, class, order, family, genus, species. I don't know. Okay? And I would know what are some of the things you use to classify organisms. What are some things you don't use to classify organisms that you have to worry about? What are the differences between convergent evolution, divergent evolution, parallel evolution? How do we know where to put things? What would be an example of in your lab where you looked at things that were similar? And what would you compare and say, oh, you're more closely related because of this or that? How do you make those determinations? It's not asking you to memorize every classification. It wants to understand as a scientist, how would you determine that? What would you think is most important, right? In order to determine those phylogenies. Then chapter 19, then we went to chapter 20. Okay, how come I don't have that on my calendar? But we did go to chapter 20. Chapter 20, was that our viruses? Okay. All right. So on that one, you want to know, we talked about viruses, so you want to know what is viral structure. We want to talk about what cycles. Lytic and lysogenic. Then we saw some other variations on the cycles, right? Animal viruses versus retroviruses. How are they the same? How are they different? How is that adapted to a virus? Then we talked about archaea. Well, we've spent a lot of time in archaea already. This was not our first rodeo with archaea. How are prokaryotes different from eukaryotes? Things like they don't have linear chromosomes, right? They don't have a nucleus. Don't forget the basic biology that you know, because half the time I will ask you those questions and you're so memorized up, you're not thinking about the big picture like, oh, of course it doesn't have that. Of course you can't pump water, right? Because some of you still missed that. One of you had a quiz, one class grade had a quiz about that, right? And that's something you've heard multiple times. So think about the differences between prokaryotes and eukaryotes. And then archaea, we looked at them, that they were the what phials? Extremophiles, right? And they are adapted to particular environments. All right? Then what came next?
23, 24.
Right, so then we started talking about plant, is that plant structure? Yeah. Okay, so then basically from there on out, it's all plants all the time. Is that accurate? Yeah. Okay, so we talked about, okay, well, what are the things plants have done? They have gone from living in the water to getting on what? Land. What are some of the hurdles they had to overcome in order to do that? What kind of relationships did they develop with other organisms in order to do that? What kind of coevolution? There's multiple examples of coevolution with plants, with other organisms, right? And look and see, well, what structures did they have to have, and why do they need those structures? What do those structures do for them? What do roots do for them? How does that allow them to be competitive on land? What part of the stem is valuable? Why is it that way? What's with the leaf? What makes the leaf adaptive? Then once you understand that, then you can look at the variations on the themes, right? Why would some leaves be broadleaves? Why would the stomata be on the bottom of the leaf? All of those. So then you need to go into specific, we talked about two things specifically about plants after we learned the structure. We talked about reproduction in plants, and we talked about movement of fluid through plants, right? So movement of fluids through plants, we had to talk about xylem and we had to talk about phloem. And those are two big ticket items as well. In xylem, we learned about the structure of xylem, and we learned it, do your Venn diagram on xylem and phloem. How are they the same? How are they different, right? And we know for sure xylem is what? Dead. And then we talked about what moves water through that plant. And what did we say? well, we said root pressure got it started, but basically it's what? Yeah, TATC. And I would know what's the T, what's the A, what's the T, what's the C. I would be able to explain that. If you had a lab over it, clearly it's important, right? So be able to explain each of those. Water potential's got to be in that. As I was reading through your labs yesterday, which is all I did, was read through your labs. And I'm almost done with 5th period. So if I know 1st and 3rd is it posted for you? Or no, 1st and 5th I'm done with, because I try to mix up the order so I'm not easier or harder on anyone. Like if I get tired and I start getting sloppy. So try really hard not to. But I think 3rd, you don't have yours yet. I'm halfway done through 3rd, so I'll finish that up and get those posted. And I think your hyperdocs, oh, you haven't seen them yet? No. Oh, okay. Just the 2nd hyperdoc you haven't seen. The 1st one you have your score on. Yeah. 1st and 3rd is done. I'm working on 5th on that one. TATC, thank you. As I was reading that, some of your hypotheses, one thing that people lost a point for was they told me that everything increased transpiration in your lab. Did everything increase transpiration?
No.
No. What decreased transpiration? This compared to the control. But all you like, oh, all of our conditions will increase transpiration. No, I don't think so. Another thing is you would write a hypothesis and you had no water potential in your hypothesis. If you at least explained it, I talked about it, you're like, it's like it's a mystery, some things affect transpiration. I'm like, really? Because they gave you a whole lecture on it. I'm curious. It's like some things happen, like, oops, dropped my pen, okay? Explain why, okay? In your lab, of course, you should do that, but on your test, you should, of course, explain that to me. Don't just use a word, oh, it's going to increase. Okay, why? Tell me why that is, right? So that was something that I saw there. So make sure you talk about water potential. We talk about phloem, it's alive. There's the companion cell. It's moving of sugars. We talked about the what hypothesis? Pressure flow hypothesis. I would understand that. How does it work, right? What are some similarities we've seen? Remember what was the one thing we saw throughout all plants was the pump. What was that?
Proton.
Yeah, it's how we got root pressure going. It's how we got stomata open. It's how we got sugars in the phloem, right? Those are all things that you can look at. Flowering plants, and you had highly suggested reading and thinking on coevolution between plants and herbivores and adaptations to protect themselves. How to message other plants. We had coevolution and pollination, right? Those were all different things that we talked about. And for reproduction for plants, you really need to make sure you're comfortable with the double fertilization and the gametophytes in each one, which one's haploid, which one's diploid. Yeah. And I think that is the unit. Is there anything that you want to ask me particular questions about that you're not feeling comfortable with? Yes?
The cation exchange capacity?
Sure. Okay, so cation, is that positive or negative? Positive. Yeah, so where we see that would be in the roots, okay? And so what you do is you could pump protons out, which that would cost you energy, right, to do that, some form of energy. In our electron transport chain, we got the energy from the electrons moving from a higher energy level to lower, right? Okay, but here you have to use ATP to move your protons out in exchange for some other ion you might need. Another thing you can do is pump protons out, and then when those protons come back in, it's +1. Its date could be a negatively charged ion. So you could use it as its escort to get it into the root. And then because of that, the critical part of that is what are you doing to the water potential inside the root? You are
Dropping.
Yeah, you're lowering the water potential in the root. Then the water will move from the root soil, the soil around the root, into the root. And then let's drop the water potential in the stele, same idea. Water comes in, apoplast or symplast, into the stele, symplast. You can't use the apoplast anymore because the endodermis has those waxy Casparian strips. Remember that? Yeah. But as you go through, so it's going to be regulated, and then it keeps moving up. You're going to have a lower water potential in the leaves, and the lowest water potential is going to be the air outside. Now, did your mist transpire at all? Your plants in the mist? Yeah, but it was really, really what? Small. because the water potential was so close, so similar, that it didn't have a gradient to change, right? So it still transpired, just not as much. Okay, what else? Yes?
Can you go over double fertilization again?
Yes, I can. So double fertilization has to do with the 7-celled gametophyte of the angiosperms. Okay, so this is the female gametophyte. It's 7 cells big. These right here are referred to as the polar nuclei. Did I spell that right?
Yeah.
Yes.
Okay, it looks funny. Okay, so, and then this right here, this is the actual egg. Okay, and this would be inside the ovary, inside of an ovule, inside an embryo sac. Russian nesting doll, right? Okay, so if I made this, let's see if I can do, oh, poop. Okay, so then if I made this inside, what am I drawing? Oh, this is, okay.
Sorry for the interruption.
No.
Kathy Schulteis, if you're at school, please come to the office. Kathy Schulteis, if you're at school, please come to the office. Thank you.
She didn't come and last week she did too.
I'm glad you're here. Okay. Okay, now come back to me. All right. So when I need a different color. Here we go. When the pollen lands on the sticky stigma, all this pollen will start growing down, right? And inside that pollen tube will actually be three nucleus, okay? 3 nuclei. One is just the tube nucleus, right? Minecraft, building down, okay? The other two, one will fertilize the egg to form your diploid zygote, right? And one will go over here, and this will then become the 3N or triploid endosperm, which will move into 1 seed leaf if it's a monocot, or 2 seed leaves if it's a eudicot. Oh, we studied that too, monocots and eudicots, right? Yeah, sure we did. Does that answer your question? So this is the double fertilization. You fertilize the polar nuclei and the female gamete.
So then what above the 3 dots? What are those?
Antipodals and synergids. Those are just other cells. No. Okay, I'm just going to left to right. Yes?
How exactly do trigger movements respond to stimuli?
Okay, so that real quick movement is probably an electrical stimulus, and that's all I would talk to you about that. Okay, I'm going to cross, yes?
I'm just making sure, if you would ask us how water moves up the sound, you would mention the TATC theory and then say water potential to support that, right?
Well, water potential is part of which part of that? Anybody know? Tension, yeah, tension. Okay, yes?
Could you please go over the complete, incomplete, and perfect flowers?
You don't need to know that. Remember I told you that. It's an itty bitty writing. Yeah. Uh-huh.
Would you be able to explain the interactions between the environmental stimuli and the internal molecular change? Or does that have to do with the hormones?
Yeah, those are well, environmental stimuli could be abiotic or biotic. So your environmental stimuli could be when the sun comes up and when the sun goes down. It could have to do with temperature and the heat, right? And whether or not you're evaporating or not. So stomatic closing is responding to an abiotic factor, or the stomatic closing is the response to a dry environment and they're losing too much water, right? It could be leaves folding at night, right? Those are all abiotic factors that you're responding to.
So would, for example, phototropism be?
Phototropism would be responding to your environment, growth towards the light, or in roots, growth away from the light. So that's responding to an environmental stimuli. Yes.
So for the polar nuclei, are they just there? Oh, is it from?
Oh, okay. Remember the mother Megaspore? The mother of dragons, right? She's not actually a Megaspore. So she undergoes meiosis, and she forms 4 spores. And 3 are dead to us, and 1 survives. That Megaspore, is it haploid or diploid? Haploid, because you just underwent meiosis, right? Meiosis. That haploid Megaspore will do mitosis, which will then form how many cells? 2. And then mitosis again, which will form 4. And mitosis again will form 8. No cleavage here. So you have 1, 2, 3, 4, 5, 6, and you could be 7, 8, but you don't, and that's the polar nuclei. Okay, I'm working across. You already got a question, so I got to keep going, and then I'll come back. Yes. Quiet, please. Shh.
Phytochrome and photoperiodism.
Okay, so phytochrome is a pigment. It's like a toggle switch. Okay, and so in the morning, its state is like this, like PR. Okay, red light. It's ready to absorb red light. Up comes the sun, and more of the phytochrome, and this is part of photoperiodism, right? It's going to change into the far red state. So towards evening and dusk, you're going to have a lot of far red. It's going to be in that state. Then throughout the night, it breaks back down to PR and starts all over again. Okay? Okay, now I'm starting over here again. Yes?
Just to clarify, besides things like the cation exchange capacity, we don't need to know any of the other itty-bitty writing?
Right. Itty-bitty is there for support. Yes?
What do we need to know about phototropin?
What is phototropin?
It's isn't it a receptor that
Mm-hmm.
absorbs blue light and gets
Mhm.
phosphorylated?
Yes.
Is that it?
That's it. Okay? Yes?
What types of roots should we be familiar with? Like, should we know food
Well,
storage, aerial roots?
I don't think they're going to ask you questions on particular types of roots, but if you heard this word, fibrous root versus taproot, which one is a carrot? Taproot. Yeah. Fibrous root. So there are different adaptations and roots for their particular environment. So you get, roots that are buttress roots because they live in a swampy environment. And you might have other roots that have, pneumatophores, which they grow back up this way because they are suffocating in their muddy and wet environment, so they need oxygen. So those are just examples of adaptations that roots might take in order to respond to their environment. Okay, I'm coming across. Yes. Do I see another hand up? Yes.
So just one thing that keeps throwing me off. Ground tissue isn't actually in the ground. It doesn't have to be in the ground.
No, it's like it's the groundworks. It's the basic self. Yeah. Yes.
Alright, this is a stupid question.
I'm sure it's not.
For the movement of sugars in the fall again, how that.
Sure. Okay. So the first thing that happens is you have a source. You would need to talk about a source and a sink, right? So what could your source be? Where the sugar originates, which could be, if it's in the spring, it could be the leaves. If it's in the winter, it might be in the roots. Right, it's wherever your sugars are stored and are getting concentrated. Your sink is wherever they're going to move to. If it's the springtime, you're getting ready to store for winter, so you're going to move it to the roots, right? Or you're moving it to the growing shoots. Yeah, so that's the sink. So the idea is you have a differential. And that differential is have a lot of sugar here, don't have very much sugar here. Why don't I have very much sugar here? I'm either using it up quickly because I'm growing, or I'm storing it into a more complex carbohydrate in the root. So that's what makes it a sink. So you have a differential, high sugar, low sugar. So now we just got to get it going, right? We've got to overcome inertia. We've got to move it from here to there. So the thing that's going to help us do that is, again, a proton pump. So use a proton gradient to escort sugars. When protons want to go back into the sieve tube, they'll bring with them a sugar molecule. So now they've moved into the phloem. Now we're going to make the inside of the phloem, because we have all those sugars in there, we're going to make it what? What would it become by default? High protonic. Okay? Water's going to follow water potential. Water must flow from the hypo, right? High water potential to low hypertonic situation. It's going to go in to that sieve element. So trigger pressure is going to go up. It's going to be trying to push things out of it. It's not going to push it back to the source because here comes more sugar, here comes more sugar. I can't go that direction. I got to go this direction towards the sink. That process just repeats itself over and over. And at the sink, you're either consuming those sugars because you're a growing plant, or you're storing those sugars as a different form, not a simple sugar, but a more complex carbohydrate. Does that make sense?
Yeah.
Okay. Next. Yes, I'm sorry.
Why do the sources change, like for storing food or something? It says that
Why do the what?
like the sources change. It says that the stored root can be a source or a sink.
Oh, because sorry. Oh, I forgot those 7. Look at the tree out here right now, right? So, right now the roots are a source because the leaves certainly aren't, because there aren't any, right? So your source of sugar is now in the roots. But when you first came to school, it was probably in the leaves, right? And that's why phloem works any direction, and that's another reason why phloem is so awesome, right? Because that's why you use it to send signals, right, through the plant, is use the phloem because it moves all directions. Okay, what else? Yes?
What is bulk flow?
Bulk flow is pressure flow hypothesis.
Okay.
You're, moving sugars in bulk. What else? Oh, I'm sorry, yes.
For the pressure flow hypothesis, the movement of sugar is co-transport, and then water is passive, right?
Well, there's lots of examples of co-transport. For pressure flow hypothesis, the only part that's co is when you just move it into the sieve tube itself, right? Because it's going to hitchhike on hydrogen. So that's the co-transport. The rest is trigger pressure.
Okay, then water is passive.
Yeah, there's no investment of the plant. I mean, side note, get the party started, right, okay? But there's no investment by the plant. You're just using evaporation out the transpiration. The actual movement of water is through a dead cell, if it's most effective. It can move through a living xylem as well, but the most effective xylem is dead. It doesn't need any cellular interaction or anything to keep it going. It's, based on the properties of water and water potential. What else? Yes?
Can you explain the trigger movements to touch other responses? A leaf will go flaccid, or if the vacuole is empty, or if the vacuole is full, then it'll expand. So like a petiole on a leaf, depending on whether if it's rigid, it's going to be more upright, it's going to be open, it's going to go flaccid, it's going to drop, like in the mimosa. Right? Didn't one of my classes look that up for me? Yeah.
So what's the difference between gram-positive and gram-negative in terms of how it actually affects the cell itself?
The only thing you have to worry about how it affects the cell is whether it stays alive or not in relationship to antibiotics. That was a very random thing, gram-positive and gram-negative, having to do with staining. And it was a way because it was like all these things are small and I can't even differentiate between them. I know, here's one way. All these take stain and all these don't. Then when they actually were able to study the way it was, it's because it's like cell membrane, cell wall, and then another cell membrane. And that's what made it gram-negative, because the wall wasn't just exposed so readily that you could stain it. So that separated them that way. Then apply it a little bit further. When you have that third barrier, right, instead of just cell membrane, cell wall, you have cell membrane, cell wall, and then like another membrane on top of it, it makes them a little more resistant to antibiotics. So that would be the concern for us. What else? Yes?
How does auxin support the embryo sac?
How does what?
Auxin, it's like help it develop.
Auxin is going to do when those cells are in there, they're going to expand. So anytime cells are going to get bigger, cytokinin, cell division, right? So you still got to have that in there. You're going to still be making more cells. But if cells are going to get bigger, if you take cells, think about, have we done anything? We've done a little bit with development. Zygote, solid ball of cells called a morula. We've gone over that, right? Or is that a different class?
Honors.
Oh, okay, I haven't taught you that yet. Okay, so you can have cells that are remember after we do mitosis, how cells are small? We've studied the cell cycle, right? And what happens during interphase, during G1? Growth. So you can have a cell double in size, right? So imagine if you're talking about 10,000 cells or 20,000 cells doubling in size, right? Auxin can help facilitate that process. Doubling in size because the same cell got bigger. Does that make sense? Okay, all right. What else? Yes?
Why use mitochondrial DNA for sequence comparisons in phylogeny?
Okay, so let's talk about that. Mitochondrial DNA only comes from one source, one line, your mother's side, right? Because sperm don't bring anything but the nucleus. So when you look at mitochondrial DNA, and also for there's that, so you can draw your line, but for closely related species, you use mitochondrial DNA because it mutates at a faster rate, so you might be able to find a difference between two species based on the rate of mutation. Does that make sense? Yeah. Okay. What else? You're welcome. All right, my friends. Listen to me. If you only have 2 hours to study, what would you do?
Review the important things.
Review the important things, right? The chapters that you're weakest on. What is the best thing at this point forward? What should you do with your time? I would do the quizzes at the back of the chapters. Right? I would do the quizzes at the back of the chapters because the bulk of your points tomorrow, right? You've already earned 35 of the points that's staying in as it is, right? So now you have 50, 65 more points to earn, right? 50 of those 65 points are coming from what?
Multiple choice.
Multiple choice. So personally what I would do is if I wasn't here for the essay review on the remaining chapters, I would listen to that I posted on YouTube. I don't know how long it was, 25 minutes or something. I don't know. I would listen to that. That covers your 15 points of essays or puts you in a better place, plus what we've talked about today. Okay? Then the rest of my time, I would go and just take multiple choice question tests because that's how you will check for understanding. Okay? If within that multiple choice test, now hopefully you've gone through all your chapters, but if you find a big gap in your learning, then sit there and read through the notes again at this point, right? If you have a big old gap in your learning. But that would be the best use of your time. And I'm assuming any homework that you have due tomorrow, you knew about it on Friday. Is that accurate?
Yes.
So it should already be done. If you're at a point tonight where you have any other homework to do other than my class, that is on you, and I'm going to tell you right now, change your ways. You are in AP Biology. You knew all your homework on Friday night because you had all your classes that are going to meet tomorrow. All of that should have been done first thing. There should be nothing on your plate tonight but getting ready tomorrow for your AP exam. You shouldn't be going to bed stressed, doing English work at 11 o'clock at night, right? All that should be done. And take a moment to ask yourself, did I set myself up for the greatest success? I am not a sprinter. I cannot run fast. I am confident any one of you could beat me. But I can tell you one thing I wouldn't do is eat a pizza before we race. Because I'm setting myself up to get sick and crampy and throw up, right? Some of you set yourselves up to have a harder time tonight. Change your ways. It doesn't do any good to cry about it now. It is as it is, right? It is as it is. All you can do now is go, how can I make best use of my time? Take your multiple choice questions as soon as you possibly can. Get started on that. Find your weakest chapters. Hit those hard. Go to bed early. Have something to eat. Take a walk. If you don't work out today, you go take a walk after dinner. Talk it through. Ask your parents. They'll love you. They're trying to keep the relationship up with you. They're worried. Say, hey, can I talk you through some things? It might, there you go. All right, make good choices. That's it. Bye. Have a piece of toast.